Until 3, 3, 2, 1, live. Okay, welcome everyone. Let me start by wishing all of you a very happy 2021. Let's see what this is going to bring us. Let me first welcome you very happily to the third webinar already of our Origin Center webinar series. Basically, Origin Center is a sort of a networking platform in the Netherlands that wants to bring together people from astronomy, chemistry, evolutionary biology to study the origins and evolution of life on Earth and in the universe. Let me start by telling you briefly about the procedure. We'll first have a 30 minutes talk by Kate. Afterwards, there will be, and people can ask questions, but mostly there will be questions at the end, and we have a lot of time for questions, so we hope we get a bit of a lively discussion. Then after the discussion, we'll close the YouTube channel, we'll close the open activities, but there will be space for students to have more direct conversation with Kate. So this is basically for junior scientists. Okay, so back to our webinar speaker. I'm super excited that we have Kate Adamala here. Actually, I was just talking to her. She told me to stress you that she's European. So I'm very happy she's proud about that. Kate is, I think there are several reasons why we invited her. One, of course, is she did really beautiful work in the direction of synthetic cells, something I'm partially working on. But also she has really spent energy in trying to build a community that's open, that's sharing, that wants to make the whole field go forward. But also what's pretty interesting, actually, I was just talking to her and she mostly sees herself as an astrobiologist. I think she's really someone who works in the spirit of our Origin Center. So I think I said enough. Oh, she's actually from the University of Minnesota, which I forgot, sorry. So I think enough about this. Well, Kate, go ahead. Look forward to your talk.
Thank you. Thank you so much for having it. Giving talks in Europe got much easier in the last year. So that was the shortest flight I ever had to take to give a talk in Europe. So welcome, everyone. Thank you for having me. I will start sharing my screen. And what I want to talk about today sounds like it's exactly in the spirit of your center, which is using synthetic biology to try to answer astrobiology problems. And most of what I'm going to talk about is how can we investigate the life of the last universal common ancestor to be in the spirit of the Origins Center. But I will also emphasize how this technology can be used to do space exploration, to not only look for life elsewhere, but also to seed life elsewhere, to terraform, to do long-term space missions. So there will be a little bit of practical applications of our technology as well. So just as an introduction, this is the problem we're all facing. We know that once there were chemicals and out of those chemicals came life. How did that happen is what we're investigating. That's what we don't know. And one of the biggest problems we're having right now is we have n that equals 1 right now to study because there is only one type of modern terrestrial life. So all of biology that's been done so far has been done on a sample size that equals 1. So that's not very scientific. If you have single sample size and you're trying to figure out some general properties of life, some general features of biosphere, it's difficult because you can't really generalize. All we know about life is from our own life, and we don't know what properties are general to life. So this is what our field is trying to help with. So this is how we used to and how we still investigate all the astrobiology problems, including the problems of the origin of life. We do in vitro experiments where there is few molecules under very controlled conditions. Those molecules mix and match, and we come up with little parts of biochemical processes. Or we look at a life cell, which is incredibly complex, and we're trying to figure out, going backwards, how did that life cell started? And obviously there are advantages and disadvantages of both of those approaches. If you work with live cells, you have a black box. You don't know what's in it. You don't know how it was made. So it's kind of hard to simplify it. If you work only with in vitro processes, only with few different molecules and try to do simple chemical evolution, it's really hard to build up complexity. It's hard to figure out how something as complex as biological life came to be. And so this is where my field comes in. We propose that synthetic cells might be the solution to at least some of those problems. In synthetic cells, you have the complexity that approaches a living cell. So there's a lot of emergent properties that I'll talk about a little later, but it's basically an organism that's complex enough that you can study living processes, but it's much simpler than a natural cell. So you can answer a lot of basic questions. You can try to think of a synthetic cell as a kind of way to simplify life with still having the chemical reaction resolution. So you can still look at every reaction, every pathway in that cell. And there's also a lot of cool applied applications of those technologies that actually pays the bills, that we can have biomedicine, we can have space medicine, we can have drug discovery, metabolic engineering done using those technologies. So that kind of helps the field move the field forward. And so for the context of this talk, you can think of a synthetic cell as a model of a last universal common ancestor. You can think of it as a very simple cell that has some but not all functions of living cells. So if we build a synthetic cell that is just complex enough to do life, but not as complex as modern terrestrial cells, then we'll be able to actually understand how those cells started, how they came to be. And our field has kind of two different approaches to doing that. I'm firmly in the top-down camp. I take things that are not living and put them back together. And at some point, I'm hoping it will start to be alive. There's also people that take modern complex cells and simplify them, take things away until they end up with something that's just barely alive and can be fully understood. None of us reached the goal yet. We don't have a living synthetic cell. But in the next hopefully 20 minutes, I'll show you where the field is right now and what are we doing. This slide shows you the main rationale for using synthetic cells. Basically, if something that's built, engineered, and already mostly dead cannot be killed, and that's the biggest advantage for us of synthetic cells, is if you study a live cell, trying to figure out how it started, what were the evolutionary processes that led to the origin of that cell, you have to de-optimize the cell a little bit. And if you de-optimize a live cell, most of the time you end up with a dead cell. If you de-optimize a synthetic cell, so build a synthetic cell that's just a little bit farther back in the evolutionary timescale, you don't end up with a dead cell because the cell's already started dead. You end up with something that's just slightly more simple. And so that lets you study all those transitional processes from non-life to life. You can basically kind of hover around that life to non-life boundary with synthetic cells as long as you want. And you can study all those processes that supposedly gave rise to life. And so notice that I still haven't told you what is a synthetic cell. Well, there's not a one kind of universal definition. To me, a synthetic cell is anything that have never been alive before. So any biochemical system that resembles life, but does not come from an unbroken line of evolution all the way back from the last universal common ancestor. So the elements of a synthetic cell came from living organisms, but there's no unbroken lineage of living cells. And kind of a more trackable definition, at least to me, is that synthetic cell is anything that's more than the sum of its parts, but it's still understandable and most of all controllable. I'm trained as a chemical engineer, so I like to control my molecules. And that's why I was not happy doing biology when I was doing kind of the classical synthetic biology, because if you work with a cell, you can't really control that cell. You can ask nicely and maybe the cell will do what you want it to do, but you can't really absolutely control every molecule in your cell. And the nice thing about synthetic cells is we can control pretty much everything in a synthetic cell. We can control things that are not controllable in normal life cells. So we can control things like size, we can control the very nature of the biochemistry of that cell. We can build a synthetic cell with metabolism that combines elements of different organisms. So we can basically go all the way back in time and build a cell that can control all the elements that right now natural cells don't let you control. And that's a nice thing about synthetic cells, is that they're so incredibly modular. Building synthetic cells, we can use elements from every domain of life, every domain of modern terrestrial life. So that lets us kind of mix and match elements and figure out what came first, what elements separated from others, what elements were crucial for the last universal common ancestor. And so here's my favorite analogy that I like to use when I talk about synthetic cells. They're basically jigsaw puzzle of life. Synthetic cells are extremely modular, so you can build them in any way you want. Okay, again, I still have not defined a synthetic cell. So here is what is a definition of a synthetic cell as understood by my lab. And please understand, this is only kind of personal definition that I obviously force onto every of my lab members, but other people might disagree with it. When we say a synthetic cell, we mean a liposomal bioreactor that's capable of performing central dogma. So it's basically a liposomal membrane made of lipids that are similar to the membranes of modern cells. And inside that membrane, there are membrane channels for communication with the outside environment. And then inside the lumen of that vesicle, there is a essentially entire central dogma. So there is a DNA genome, there is a transcription machinery and a translation machinery. So we make proteins from DNA templates. The membrane is kind of relatively boring part of it. It's simple bilayer membrane that we make depending on a project, either from phospholipids to mimic more advanced cells or from fatty acids to mimic the very early primordial cells. There's also another way of doing it, which we just recently started, which is using microfluidics, where you have those oil droplet emulsions without a membrane, but with higher uniformity and higher throughput. So this is the container. What really is the interesting part to me is what goes inside the container, and that is the central dogma. That is the protein expression system. So this is a little bit more of a classical synthetic biology. You can make a cell-free protein expression system by purifying the guts of pretty much every organism. We mostly use bacterial systems because E. coli grows nicely. It gives up its ribosomes without a fight. So you can make the cell-free protein expression system from E. coli relatively easily. But you can also make cell-free protein expression systems from pretty much any type of cells, and that's been done. And for different applications, you might want to use cell-free expression systems from different organisms for different purposes. When we explore last universal common ancestor and astrobiology applications, obviously we're more towards the simple prokaryotic end of that spectrum. So we want to take very simple metabolism, very simple ribosomes. But for some applications, we actually want to use more complex ribosomes, and including prokaryotic all the way up to a human cell-derived ribosomes. So what my lab is doing is we're chemists that pretend to be biologists. We try to put together molecules until they resemble a cell, but still keeping all the kind of cellular components, keeping everything that's as simple as a chemical system, but behaves a little bit like a natural cell. And this is a diagram that shows kind of overview of what we're interested in. I love this feedback between synthetic cell engineering and tool development and understanding of natural biology. This is the kind of origins astrobiology part. If we want to build a cell from scratch, we have to understand how a cell works. But once we build a cell from scratch, we'll have a lot of tools and a much more understanding of how a natural cell can actually work. There's also a lot of kind of specific applications, including origins of life and astrobiology, space exploration. All those applications allow us to kind of look at life not as just a single phenomena that we know as a terrestrial life, but also kind of as a general phenomena. And now I'll show you examples of a few different projects, and I apologize in advance that I'm going to go pretty fast through those projects. This is just to give you an overview of what kind of things we can do with a synthetic cell. If you're interested in any experimental details of any of those projects, I'm happy to talk more later. So one of the things that life loves to do, obviously, is to breed, mate, and produce progeny. And so we developed a synthetic cell system that mimics that process. So we have a synthetic cell system that allows our synthetic cells to mate, produce a daughter cells, and then in the process of that mating, develop different lineages. So we have this one synthetic cell that we called a pluripotent synthetic cell, and that synthetic cell can differentiate into a lot of different kinds of synthetic cells, containing different kinds of genes or different kinds of small molecules. And the way it's done is by sequential fusion of synthetic cells. How do you do fusion of synthetic cells? Well, you use natural property of membranes, which is that if you drag two different phospholipid membranes together, they're going to fuse. And that's what our synthetic cells are doing. We're dragging membranes together, and that's why it's programmable, because we're doing the dragging using DNA tags. And then once the membranes are close together, they fuse, and that's followed by a lumen fusion. And so we can fuse synthetic cells in a programmable way. And we've now done it with up to 9 different populations of synthetic cells that we can fuse in a controllable way. And why we're doing it is, I'll show you, this is a very simple proof of principle experiment that lets us build a very complex genetic circuit. The identity of specific genes on this graph is not really as crucial as the fact that we can fuse a lot of different populations of synthetic cells here over 5 generations. We're fusing those synthetic cells, and at the end, we have a single reporter protein that's only possible if all populations of all 10 different genes from 9 different populations of synthetic cells over 5 generations got combined together in that daughter or of the kind of population of end population. And so only if all those parental pluripotent populations get fused, you end up with a final population. And obviously on this scheme, you see a single cell, but it's never a single cell. It's also always populations of cells. So that final population of a cell that expresses a, in this case, firefly luciferase, which is our reporter gene. None of that is living. I know some people claim that if you reach enough complexity, even without self-replication, you end up with a system that's living. I disagree with that. I think all those systems are still dead, but they're very complex for a dead thing. They can express proteins, they can undergo differentiation, speciation in this case. And what we can do is we can build those systems as complex as we want. So here's another example of how we can base, oops, I don't know why my slides jumped back. Let me go back. Another example, yeah, this. Another example of what we can do with the system is we can build synthetic cell populations that are non-linear. We can build pathways that are basically as complex or approaching complexity of natural pathways. We can, starting with one population, we can, depending on the path which those cells take, we can differentiate into different kind of those daughter or progeny populations. And in this graph, again, identity of the genes themselves, it doesn't really matter as much as it shows you that you can take a single population and differentiate it into 2 different populations, depending on who they mate with. So depending on what kinds of cells fuse at any given time. And the take-home message from this is that we can make cells to differentiate into, in this case, 2 populations. We've also shown 3 populations are possible from a single ancestral pluripotent synthetic cell. And obviously I'm using the pluripotent in a very loose way. I'm saying pluripotent as in it can become any other population, but it's not living, it's not yet living. So this is an work in progress. This shows that you can take synthetic cells and do things with them that you would normally have a really hard time doing with kind of much simpler biochemical systems, which is differentiated, make complex genetic pathways. Another thing that you can do with synthetic cells, and this is going to be the familiar graph that I've shown you earlier, is you can evolve things that were not meant to be evolved in live cells. In this case, it's a ribosome. So we have several projects where we're working on either evolution or de-evolution of a ribosome. And the reason why this is important is because the ribosome, the kind of a heart of a central dogma, we know that that's been set at the last universal common ancestor. So we know that at the point of the origin of life, ribosome was already functioning. The peptidyltransferase center and some of the surrounding proteins already existed. And it have not changed in the last 4 billion years. So since the nature figured out how to do protein synthesis using this ribozyme that's packed as a ribosome, it never went back. There is no mutations in the core, in the catalytic core of the peptidyltransferase center. So we can't really mutate that in vivo, but we can mutate that in synthetic cells. And that's what we're doing. We're mutating parts of the ribosome that have not been, that we were not able to mutate in live cells. And one of those parts is the ribosomal hinges, which control the head swivel, the motion that propels the ribosome to move on to the next codon. So that head swivel of the ribosome, like the catalytic core of the ribosome itself, is guided by the RNA, by the RNA hinges. And so controlling those hinges is one of the things that we need to do to change the processivity of the ribosome and to look at the ribosome as it went back, as it's kind of how it started. And so we're doing that using in vitro protein synthesis. We're doing that using protein synthesis, using tools, modern chemical biology tools, to try to see how a ribosome looked like at the time of the last universal common ancestor. And I realize this slide is kind of pretty complex, but essentially what it is it's showing you a selection scheme to put a broken ribosome, broken as in de-evolved ribosome, in a synthetic cell, and then try to see if we can pick even a little more broken but more ancestral ribosome out of that mixture. I'm happy to talk more about it, but I'll move on in the sake of time, just to kind of give you an overview of what is possible to be done. And if you're interested in this project, again, I'm happy to talk more. Another thing that we can do with synthetic cells is we can synthesize things inside synthetic cells and then export them to the outside. We do that using cell-penetrating peptide tags. So we synthesize proteins inside the synthetic cell, and then we export them into the outside. We do that with those cell-penetrating peptide tags that are attached to the proteins that we want to export out. And so what you see on the scheme is a size exclusion purification traces of synthetic cells, which are essentially liposomes. So this is liposomal purification in two different channels. The top is fluorescence of the membrane. So it shows you where the cells themselves are. And the bottom is fluorescent of the protein, in this case, green fluorescent protein. And so with cell-penetrating peptide tags, after making those proteins inside the synthetic cell, most of them get exported outside of a synthetic cell. So if you purify that population of synthetic cells, you can see a sharp peak for where the membrane is, and then most of the protein is outside of the membrane. So you can basically see that with cell-penetrating peptide tags, the membrane is still intact, but the proteins get exported out of a synthetic cell. And in the absence of the cell-penetrating peptide tags, all of the protein overlaps exactly with the peak for the membrane. So that means that all of the protein still sits inside a synthetic cell. What is it good for other than making pretty graphs? For one, it's good for exporting useful enzymes. So for example, here I'm showing that we have exported a protein carrying a payload. So we basically built a protein that binds to the RNA, and that protein is tagged with a cell-penetrating peptide tag, and that protein can export a payload of a RNA, in this case, a short fluorescent RNA aptamer, out of the synthetic cell. If we can export things out of synthetic cells, we can also import them into another population of synthetic cells. And in this case, we can export a T7 RNA polymerase from one population and import it into another population that then starts expressing a reporter gene. Again, I'm happy to talk about details more, but the take-home message is that you can use synthetic cells to make proteins and other things inside and then export it and import it into another population. This would be a great way to study how origin of populations, how origin of speciation happens. You can imagine mimicking very early population divisions where there's division of labor between two different or three different or more populations of cells, and they exchange either proteins or small molecule reagents. That's also possible to mimic sort of an origin of a nucleus or other organelles. If you have one population of synthetic cells that's capable of exporting something in, and then you put it inside another larger synthetic cell, you can imagine studying how organelles came to be. I know this is late in the origin of life to the origin of life party, because obviously we waited quite a while till we discovered organelles, we as a biosphere, but origin of organelles can also be studied not just as origin of eukaryotic organelles, but also origin of prokaryotic organelles. Prokaryotes have organelles, which a lot of people don't think about. They have membraneless organelles. They have P-bodies, not really P-bodies. They have those membraneless kind of coacervates in the cytoplasm that are used to kind of sequester reactions, but we don't know much about them because obviously they're super hard to study. So this is an experimental system that let us study some of those things. And here's another thing is we can co-culture synthetic cells with natural cells. So we can translate signals from the environment using synthetic cells. And what this poor resolution picture shows you is co-culture of red synthetic cells with green natural cells. I'll show you one of the applications of this co-culture. So one of them is, can you combine talking between synthetic cells and natural cells and trying to evolve organelles? And you can combine that in the form of building artificial organelles. So here's an example of a natural cell, a cyanobacteria, encapsulated inside a synthetic cell. Now you're asking, why would we do that? Well, for one, because natural cells could be really good models of artificial organelles. And one of the things that that artificial organelle can do is build lipids, provide lipids so that the synthetic cell can grow. And what you see on this video is a synthetic cell growing as a result of lipid synthesis from a natural cell. So that synthetic cell has a natural cell encapsulated inside it, and that cyanobacteria produces lipids, and the synthetic cell grows into those kind of threads. And another thing you can do with those populations is look at the evolution of traits that propagate across populations. And one of the kind of easiest to imagine traits that propagate across populations of very simple early cells is resistance to environmental stressors. And one of the environmental stressors that we study is an antibiotic. This also has obviously practical applications towards studying emergence of antibiotic resistance in modern cells, and especially those multi-antibiotic resistance infections that are starting to plague our hospitals. And so what we do is we have two populations of synthetic cells. One of them has a gene that provides resistance to antibiotic, and the other one doesn't. And this is just a very simple summary of the types of experiments you can do with this system. On those microscopy images, you see, again, red is membrane, green is representation of a synthetic cell metabolism approximated by synthesis of a green fluorescent protein, reporter protein. And so in presence of antibiotic and antibiotic resistance gene, you see that membrane is obviously intact, but also metabolism is intact because those cells still synthesize their GFP. Without antibiotic resistance gene in presence of antibiotic, there is no metabolism inside cells. So the antibiotic doesn't disrupt the membrane, but it does mess up the metabolism to the point where you can't detect any expression from the proteins. And so again, I'm happy to talk more. That's just showing you that it's possible to mimic things like antibiotic resistance in synthetic cell populations. Another thing we're trying to do is we're trying to put together a synthetic cell from a simplest known living cell. So JCVI has this mycoplasma-based synthetic cell that's called SYN 3.0, which is a simplest known genome of a natural living cell. And that genome is basically the smallest known genome of an independently living organism right now. And we're taking that genome and putting it into a synthetic cell. This is work in progress that I'm only showing to show you what's possible. It's possible to imagine taking a DNA of a smallest living cell, putting it into a completely non-living cytoplasm, and hopefully at the end of it, that cytoplasm will start producing all the proteins and will restart the actual cell cycle. Another cool thing about synthetic cells is because they're synthetic, you can digitize the information about making them. And if you can digitize information about making something, you can send that information to a remote location. And that allows us not only studying biology by sending information about how to make specific samples, but it also lets us send information about specific samples to a really remote location. This is where I started. This is where this talk is going to veer into this kind of a weird areas. We're going to talk about space exploration and using synthetic cells to figure out how life started on other worlds, but also to send life elsewhere. And so an example of a project that lets us study how life started on other worlds is building this so-called liphometer. So we're building synthetic cells of increased complexity, and we're analyzing those cells to see what is the cutoff for life. So basically, at which point a non-living cell becomes living. And the idea for it is that we want a quantitative way of telling if a biochemistry is alive or not. And the reason we need it is because once we get samples back from any other body in the solar system, and we will get samples returned from Mars, we'll get samples returned from other places. There are missions to pick up samples from Enceladus. There is a mission in planning to drill under the ice on Europa. We're going to pick up organic samples, but they will be dead by the time we pick them up. There's no way a sample from, let's say, Mars or Enceladus, sample of, even if there is actual living cells there, there's no way that sample will be kept alive by the time we get our hands on it. The thing about looking at a dead organic soup is that it looks dead to you, right? And so it's really hard to figure out if it's ever been alive. So imagine you got a sample of organics from under the ice of Europa. We know there is organics on Europa. We know there's organics of Enceladus because we've done spectroscopy to know that. And so we bring a sample, it looks dead. How do you know if it's ever been alive? And so that's the goal of this Lithometer project. We want to find a way to analyze increasingly complex organic molecules in a way that helps us to decide if that sample is coming from just abiotic processes or from something that has ever been alive. And so kind of segue into going into space, actually. Can synthetic cells go into space? And if I'm showing it to you, then you already figure the answer is yes. We were privileged to be part of this amazing team of people that decided let's send synthetic cells into space. And so synthetic cells, in our understanding, were extremely simple at that point. We wanted to put two different functional types of synthetic cell biochemistry on a rocket, send it up and see if it survives. So if you think about measuring metabolism of synthetic cells, you've seen that on my slides before, you want to make GFP. Green fluorescent protein, which is this beloved reporter protein of all of synthetic biology. And so that's what I designed when I started working on this project. Unfortunately, GFP, which I didn't think about before, requires oxygen to maturate. And under those conditions, under those sounding rocket conditions, we were not sure how much oxygen we're going to have. So we decided to build a simpler system and test two different things. We decided to test whether RNA can fold into functional domains in a synthetic cell system under spaceflight conditions. And we decided to check if we can express some proteins. And in this case, a very simple reporter protein, a protein called FLASH, which is spectrally similar to GFP, but it's much simpler. And so we put those two experiments, those two cell-free protein expression experiments, we loaded it up on the rocket. And then I'm going to show you a video that makes me super happy and super proud, because this is the first time ever that I've seen my own payload go up on a rocket, blow up into space. So I was obviously really happy. It blew up into space, it came back, we recovered the sample, we analyzed the sample. Now we can say that it is possible to put synthetic cells through this whole launch protocol, get it assembled, build it. Now what we're trying to do is we're sending up a sample for actual human astronauts to manipulate it and to do much more complex experiments of synthetic cells that are actually able to interact with natural cells. And so this is our project that we called SCRAM, because NASA likes cute acronyms. And the idea is that we build synthetic cells that interact with natural cells, with this layer of sensors being synthetic cells and the layer of actuators being natural cells expressing reporters that tell us if it worked or not. And we build this system where we use synthetic cells to control natural cell populations. And that applications for that are not only in kind of figuring out how complex synthetic cells can be, how complex synthetic cells can communicate with natural cells, but there's also a lot of practical applications, including making medicine, making small molecules for long-term space missions. So this is the segue to the actual human astrobiology that I promised. Once we send people into places like Mars, they're going to be away from the whole support system, terrestrial support system, for a few years. So we need to find a way that to make everything they need, including things we don't know they will need in advance, using this biochemistry, using this kind of a biochemical system for synthesis of molecules in a programmable, engineerable way. And that's where synthetic cells come in. And just at the very end, I would like to show you that there is this amazing, vibrant community of people that engineer cells. You probably are very familiar with it because the Netherlands is one of the I think it's one of the countries where there is the highest concentration of synthetic cell engineers per population. You have basically synthetic cell engineers wherever you throw a rock. At most universities, you will have someone who's amazing and well known in our field. Every time I think about it, I think maybe I should have looked for faculty jobs in the Netherlands instead, because there's just such an amazing community. But there's also amazing communities of synthetic cell engineers elsewhere in the solar system. And that includes places like South America, where there's actually a babies build a cell organization right now, started in Brazil. There's a lot of us here in North America. There is a very vibrant community in Asia, especially Japan being kind of leader in synthetic cell engineering. So there's a lot of us all over the world working on engineering synthetic cells. And if you're interested in any of that, I invite you to check it out. Check out our website. We have weekly seminars. We have biannual meetings. We have a lot of working groups that always look for new members. If you want to be part of it, if you want to be part of this international open collaboration that thinks about how to make cells from scratch for all different purposes, please come join us. And I want to thank you for inviting me. Thank you for listening to my talk. I want to thank everyone in my lab who made possible all those experiments and all those results that I've been showing for the last half an hour. And I want to thank all our funder support. And thank you very much. And if there's any questions, I would be happy to take it.
Yeah, thank you very much. I mean, I'm just going to applaud.
Thank you.
It was super exciting. Yeah.
And I apologize if in the middle of my talk you heard dog barking. My dog did not like me talking, so he was barking in the background.
I think it was too exciting to be bothered. Okay, maybe what I would ask is whether the people who are here, whether those who feel comfortable, whether they can switch on their camera, because it sort of feels a little bit more like an audience. And then I'm curious whether who would like to start asking a question. I mean, I could start because I was triggered actually by several of your slides.
Shall I start with a question from the audience on YouTube otherwise?
Yeah, there's two questions I also saw on YouTube, so why don't you, please go ahead.
Okay, so there's a question from Kees Dekker. He says, interesting that you call those fused complex liposomes dead and not living. Perhaps I missed it, but can you give a clear definition of life? When would you call that complex liposome alive?
Thanks, Cass. That's a curveball. I will not give a definition of life because I don't think we have a good one right now. I can tell you what's my personal definition of life. And my personal definition of life is it has to self-replicate and actively maintain homeostasis. And our synthetic cells do maintain homeostasis, but they do not self-replicate. So there's those generations of synthetic cells are being made, but they don't self-propagate. So if you leave them on a bench and even if you feed them, they will not keep making little baby synthetic cells. They will just sit there and eventually die. Or wait, I shouldn't say die because I said they're not alive. They will eventually decay. So to me, that system is not alive because we don't yet have self-replication. That's my personal line that I draw. There are people that argue that these are already complex enough to be living. And I obviously respect the right to say so, but I think they're not alive because they can't yet undergo Darwinian evolution spontaneously. So they can't replicate their genome spontaneously and they can't divide spontaneously. I would love to be proven wrong. I would love for someone to actually show that these are completely living. That would make me super happy, but I think they're not living yet. We're getting there, definitely. I think within the next 5 to 10 years, we'll be able to say that we actually did engineer life in the lab, but I don't think I'm there yet.
Okay, I think at least you gave a clear answer. He says, thanks for answering. Self-replication systems that exhibit homeostasis, very clear.
And I know that I'll be called back to that. Like I hate making definitions because anytime you make a definition, then people come back and that comes back to haunt you because you'll build something and you change your definition and people say, oh, you used to say something else. But I think that's a problem of our entire field. I would be very curious to see, for example, what Kes has as his definition, because the systems he's building are incredibly complex too. And especially this microfluidic magic that he does, build cells that are so complex, so reproducible, that they, if you look at videos from his papers, it looks like they're living. So I can see that where that's coming from.
Yeah, thanks. I mean, there are more questions in the YouTube channel, but I'm also curious whether there's someone here in the audience who would like to ask questions. Okay, Reza, go ahead.
Yeah, thank you, Kate, for your interesting talk.
I've
been curious because here in BASIC in the Netherlands, we in our project we have kind of different, pathway or a different way to approach the synthetic cell. We are actually kind of building engineering without taking the application into account that much. But I wanted to know whether the way that you are approaching that, like what I saw in your talk was like you're taking in, like you're looking at the applications much more than what we do. And I want to know whether this is like a pathway to approach and at the end have a synthetic cell, or it's just you're kind of away from building a synthetic cell, so you decide to look at the applications first and then something like that.
The answer is twofold. One is I have to pay the bills, and people that have money want to see results, and so it's much easier to get applied science funded, at least in the US, than complete basic foundational research. And so most of my funding is coming from people that want to see synthetic cells used as tools to do X or Y. And so I'm trying to give them X or Y, but on the way to that, I'm also advancing the science of synthetic cells itself. And so that's kind of a roundabout way of saying I'm doing foundational science because it's needed to get applications. But also, I think our field needs to grow. Right now, we have a few dozen labs all over the world that do specifically synthetic cell engineering. And from the perspective of Netherlands, it sounds like this is vibrant field because every other person and their uncle does synthetic cell engineering, but that's not true worldwide. There's a lot of people that still think what we're doing is completely crazy. And I think applications is the way to show people that this is not just a theoretical considerations or foundational science. I think if we can actually show synthetic cells that do useful things, for example, that antibiotic resistance project that was born from a collaboration I have with clinicians, with people that actually treat patients. And so the idea is to show that synthetic cell technology can actually be useful for real life applications. And I think that could bring influx of interest, people, PIs, labs, also funding sources into our field to let us grow. Let us become an actual robust field that works on those foundational problems, but also has practical applications. That's why I think it's one of the kind of most appealing areas of astrobiology for me, because it's an area that actually has consequences in real life, applications in real life, and it's easy to convince people why we're doing it, why we're using all those resources on building things that not only answer those big fundamental questions like where did life come from, how did life start, is there life on Mars, but it also answers a question of how to make a personalized dose of medicine for a cancer patient or how to fight antibiotic resistance. So that's, to me, making applications is fun in and out of itself, but it's also a tool that we use to convince people that what we're doing is not that strange and not that crazy and it's worth supporting. I don't think I need to convince anyone on this call why it's worth building a synthetic cell, but you'd be surprised how many people think we have a perfectly good functional living cell. Why do we bother building another one?
Right. Yeah, I think it's very important to make that point. Yeah. Christina, would you like to ask a question?
Yeah, a bit in the vein of your answer, actually, and related to the astrobiology kind of side of your work. You were mentioning this kind of project where you say, okay, we can combine like bacteria and synthetic cells to make bespoke molecules for Mars mission, for example. So a bit of a devil advocate question: why not just send bacteria and DNA into space? Why is there the need for having this extra vehicle or extra complexity with a synthetic cell? Just curious about your answer to that.
Thank you very much for that question. That is a very good question, and the answer is because bacteria are too stubborn. So basically, terrestrial life that evolved, on Earth is capable of surviving pretty extreme conditions by our standards, but those extreme conditions by our standards are nothing compared to what's out there. For example, when you want to colonize Mars, if you want to farm anything on Mars, we obviously all watched a movie where a guy grows potatoes on Mars. That's not going to work, and not for the reason that potatoes won't grow from seedlings like that, but because Martian regolith is full of this compound called perchlorate, which is a strong oxidizer, and it's lethal to all known terrestrial life. So we don't have extremophiles on Earth right now that would survive in high perchlorate soil. And so if we want to colonize Mars, we need to find a way to make biochemistry that will survive in hyperchlorate. And no natural terrestrial bacteria can survive in hyperchlorate. There is not even a pathway that we can think of to create an extremophile of that kind. And then on top of that, you have to think about it, they will have to be polyextremophiles. So they won't only have to deal with perchlorate, but they'll have to deal with temperature variations that are unique, that are way outside of the boundaries of temperature variations that we have on Earth. And also they'll have to survive very low humidity, very low partial pressure, at least at the beginning. So we have to build basically a terminator of all organisms, a very specialized but very far out of the current capabilities of terrestrial extremophiles. And so
because maybe now I want to interrupt, because so far all the synthetic cell stuff that we've been building is based on existing genes.
Yep.
If there's no genes that does any of this, I somehow don't see how a synthetic cell based on existing genes is going to solve your problems.
It's not going to. We have to develop new biochemical pathways to do that. We won't be able to do that using existing genomes. We'll have to, and that's kind of this jigsaw puzzle part of the synthetic cells. There are enzymes in nature that can catalyze the transformations we need to survive under those conditions, but they don't exist as one unified gene pathway in any organism on Earth right now. So we basically have to learn how to mix and match genes that come from a lot of different organisms to build a synthetic cell that can actually be that polyextremophile. And then that's coming back to Christina's question, then we stick a natural cell inside it if we want to, because natural cells are great at doing what they evolved to do. So if we, for example, want to have photosynthesis, we can either build artificial photosynthesis, and people like Toby Erb are doing a lot in that direction, or we can just take an organism that's paid to do photosynthesis for a living, like a cyanobacteria, stick it inside a synthetic cell, and then just extract the nutrients. But the outside shell has to be engineered, and that's where the synthetic cell comes in. We have to build those gene pathways. We don't have them right now, and I have no idea how to build them because I'm not an extremophile biologist. We have to make a synthetic cell that is capable of accepting genes from a lot of different sources, and that those sources can build a new pathway that will eventually be able to do what we need to do to build those polyextremophiles. I'm sorry, I don't have a kind of a specific answer. I can't tell you which of those genes there will be because I have no idea. I don't think anyone knows yet. But we at least can see a path. We can see that if we get better at engineering synthetic cells, then we can start to build them up in complexity and build more genes into them that will do what we want them to do.
Yeah, thanks.
Will we go back to our YouTube questions?
Yeah, I have a question here from Ali Nick Maleki, and he asks, related to last universal common ancestor, could this be transition from non-cellular to cellular replicators created multiple origins of life?
That is definitely one of the things that we can investigate with this technology, the transition from non-cellular to cellular. Most of the work that my lab is doing is already at the individual cellular stage because we encapsulate it into the membrane. But there's definitely people that are in kind of the sphere of synthetic cell engineering that don't encapsulate. And they look at replicators, replicators in either microfluidics or replicators in completely in bulk, and then trying to look at emergence of individuality and emergence of compartmentalization in that. And I really like the mention of multiple origins, because a lot of the time people talk about the origin of life, like it happened and it was once and done. And that's probably not how it was, obviously. We had multiple origins and we just happened to have one that survived. So studying that would be really fascinating.
Thank you. And there's another one from Anthony Muller who asks, do your cells have an energy source?
They do. We have energy regeneration system that starts from the compound called 3-PGA, which is one of the natural Krebs cycle compounds. And so we do regenerate energy, but in a limited way. So our synthetic cells can go on for, without adding additional energy, 24 to 48 hours. With adding additional energy, they can go on for as long as the ribosomes stay viable, which is about a week. And that's the extent of our energy regeneration right now. And that's going back to the earlier question from Cass. That's one of the reasons why I don't think they're alive, because they need to be spoon-fed basically a high energy molecule that can be used directly to make ATP. They don't have any specific energy homeostasis mechanism to really sustain themselves.
Yeah, thanks. Cecilia, did you have a question?
Oh, yes. Thank you for such a lovely talk. My question don't make sense, but I still want to understand that thing. You had said that like it was in one of your slides with which is like origin of life, where you had defined the synthetic cell. And I think you said that when you de-optimize it, it mostly decay, like dying, but decayed. Did you ever try to learn something from the decay process? Because in chemistry, decay defines many things. So maybe in biochemistry as well. Or no?
Yeah. No, thank you very much. That's a very good question. Yes, we try to learn from those decay processes. This is a very loose use of the word decay. For me, decay process in a synthetic cell means basically my metabolism is dead, which means I don't see protein production. And we try to learn from that, especially in the ribosome evolution project. We try to learn how can we de-optimize a ribosome and still have that ribosome do at least few peptide bond formations. And so that's on the kind of mechanistic level. We're learning how to build a ribosome circumventing those kind of local minima of evolution that ribosome got stuck on at the last universal common ancestor level. From other perspectives, we're also looking at energy metabolism, and we're looking at gene replication. These are the two areas right now that we're looking at actively in trying to figure out how can you take that apart and rebuild it as a better system. So for example, for gene replication, we're using simple enzymes to replicate a synthetic cell genome, and we're looking at exactly what kind of DNA products we're getting after each round of replication to see how can we build a better self-replicating genome of a synthetic cell. So that's what we're learning from the decay. Another great lesson in learning from broken systems is that that mycoplasma synthetic cell project that I very quickly mentioned. So we're basically taking a JCVI synthetic living cell, and we're taking it apart to try to do a protein expression system from that mycoplasma cell to boot it back up to rebuild the life. And in the process of working on that, we learned a ton about why cell-free protein expression systems get broken, because we basically had 2 years of struggling with this project where we had a broken, dead protein expression system, and we were taking it apart protein by protein to see why it was broken. And in the process of that, we learned a lot about what breaks it. We still haven't fixed it, but we learned why it's broken. And so that's kind of lessons we're trying to learn from breaking something.
Thanks.
Thank you.
We have another YouTube question.
Yeah, there's a YouTube question from Yuvan Michaelo Grimaud, and asking, can you control the size of artificial cells over multiple generations?
Yes, you can by a very simple process called extrusion. So you take your synthetic cells and you squish them through a membrane with a certain size of pores. And so you can bring them down after each generation, you can bring them down to a kind of smaller size. In our case, that's about a micron. So you can't endogenously control the size. Once the fusion happens, they just end up really big, but you can using grad student assisted control. Basically, you have a student sitting at the bench and extruding those vesicles to get them to the smaller size. That's the only thing we can do right now.
Is there any other question from the audience here?
I have a short question. What is the size of the, I don't know, I didn't really get it. Do you use droplets or vesicles, and what are the sizes? That's the question.
We mostly use vesicles, and they're anywhere between 500 nanometers and a micron in most experiments. In the fusion experiment where they mate, they obviously get bigger, but they also get into funky shapes. If you look at the pictures of those fused vesicles, they kind of look like they look like budding yeast almost, because they're not nice and spherical. They're kind of weird and blobby. But in most of those experiments, they're nice spherical vesicles of approximately a micron. I like it to be about a micron because it's still small enough to be uniform, but big enough that I can take pictures.
Actually, I also want to ask a question. I know I'm the chair, but I was fascinated by you had this slide in there about de-optimized systems, right? And then you had the cell on one hand and the synthetic cell on the other hand, and they had the cell as basically this very short decay rate, and then it's dead. And then you had basically the synthetic cell has this very long decay rate. And I was fascinated because I would have drawn it exactly the opposite way. So therefore, I was triggered by this slide, because I would say in a synthetic cell, every component, because you try to minimal it and make it controllable, every component is super important. And I guess a living cell is no matter what trying to survive. So we know that if we take even this sort of cells that they reduced, typically a cell has like, I don't know how many, 6,000, 7,000 genes, and we only need 200, right? So I would have drawn exactly the opposite. Like in a cell, you can keep removing things and it keeps being alive. And in a synthetic cell, I think anything you remove and it's like the end of the synthetic cell. So I was curious to hear how you want to defend that picture.
So what you mentioned is about taking cells apart. What I meant is about putting external stressors on a cell. So you basically de-optimize it by breaking one part of it. So for example, in a natural cell, if you start heating up a bacteria cell, let's say E. coli, take an E. coli cell, start heating it up, you hit about 40, you're dead. And if you're dead, you're dead. There's nothing else you can do about it. There's no metabolism, you're just dead. With a synthetic cell, if you start heating it up, your protein expression rate decays, but it's still going. It's not completely dead. And that's what I mean as de-optimized. And that's what when I was answering the earlier question, what I talk about looking at results of a de-optimized system. Same with a ribosome. With a synthetic cell, if you mutate anything in RNA hinges or in a peptidyltransferase center, a bacteria is going to get dead because it just can't make proteins. In a synthetic cell, that very mutated ribosome will still work. It won't work enough to make proteins that are needed for metabolism, but it will work enough to make a tiny little reporter protein that we can then analyze. So the kind of the boundary of what we consider useful is broader with synthetic cells, because you can have a lot of systems that just sort of barely work. It's not much to write home about, but it works. And in a live cell, if a live cell gets dead, it's dead. It's done. And so on one hand, you're right that live cells are much more robust because they adapt, they can withstand adverse conditions. But once the conditions are too much, you have this precipitous drop onto a dead cell. In synthetic cells, they're almost like a jello versus a brick. You can hit a brick many times, but once you hit it once too hard and a brick shatters, you have no brick anymore. With a jello, you can keep hitting it and it's just going to bounce back. It's not very sturdy, but it's kind of bendy and pliable. And so to me, live cells are bricks. You can keep hitting it and they will just sit there, but eventually you hit too hard and you end up with dead live cell or a shattered brick. With synthetic cells, they're kind of like Jello. You can keep hitting a Jello with a hammer and you're just going to splash Jello all around, but you will still have a glob of Jello. And that's the same analogy for synthetic cells. You can de-optimize them and you will end up with a system that's worse and worse and worse, but it will still be there. It will still do at least a little bit of activity.
Maybe we don't have time for more discussion, but I will continue for more discussion because I'm not sure I agree. I guess in synthetic cell you reveal more of the innate biochemistry because you have less redundancy, but I'm curious if the final point hitting 0 is further up. But let's not continue there because there's one more question.
I would like to talk more about that, yeah.
Yes, this more because I find this very fascinating, but I'm not yet convinced. But there's one final question from the YouTube, and that is basically, Ingelus, can you ask that?
Yeah, there's one question from Jan Lutje who asks, would you consider a virus, very appropriate at this time, would you consider virus particles as living as they are able to adapt to their environment and to replicate if inside a host?
I don't think viruses are living, but I'm not an authority on that. Viruses are biomarkers for sure to me. A biosphere that has viruses has to be a living, because viruses need a living cell to replicate, but they're not alive themselves, at least for me. And I'm happy to be proven wrong.
Okay, well, thank you very much, I think.
That was it.
Are there any more questions from here?
I do have a question. Actually, I have two questions. So one is, did your work provide any evidence to any particular theory for the origin of life? Any idea you have about that? And the second one is, so you created these vesicles to have the synthetic life. Can you talk a bit more about how you made those vesicles, the production part? So yeah, those are the two questions.
Thank you. So answering the first question, I'm very biased, obviously, but I am a strong opponent of any single pure origin of life definition. So I don't think there was such thing as pure RNA world or pure metabolism protein first world. I think the prebiotic evolution was co-evolution of everything. So RNA was the catalytic molecule at one time, but did it with help of proteins. Proteins created metabolism, but with the help of RNA. So I think I'm basically the kitchen sink origin of life theory person. I think all of the components of modern life had to co-evolve. It wasn't that it was a pure RNA world where RNA did everything and was the only molecule, and then suddenly proteins came by and took over. That just doesn't sit well with me, and I can talk about some experimental work that we and other people did that shows that it might have been co-evolution rather than pure this or that origin of life. But obviously, we only have as much evidence as we create by experiments, and experiments are biased by who's setting them up. So unless we send a time probe back and actually sample the prebiotic ocean, we won't really be able to tell. Your second question, making liposomes is a relatively technically simple process. If you have lipids of certain concentration at a buffer of certain pH, they will form by layers. That's a nice property of lipids. Is obviously the devil is in the details, like always. So to make synthetic cells that encapsulate protein expression system, you have to work harder. You have to use either microfluidic systems or some water, oil, emulsion, liposome formation methods. But essentially, if you have lipids in aqueous solution at the correct pH, and for phospholipids, that pH is very broad. You can have liposomes over several units of pH, really. The liposomes will form, and they will form around whatever's in a solution. So the lumen will basically be a sampling of a solution. So it will encapsulate whatever is available in a solution at the time.
Okay, thank you.
Thank you.
Well, Kate, I think however sad it is, I think we have to end this now. I think it was a tremendously interesting discussion. I also thank the people in the YouTube channel for their intriguing questions they asked. So with that, I would like to end this webinar series but please keep in mind that there's going to be next month there's going to be another webinar from the Origin Center. I actually don't know now. Do you know what about English? Oh, actually, I forgot. But anyway,
Sorry, I just came out of vacation, so a little. But that's too far in the future.
But it's going to be equally exciting as Case Talk.
Yes, absolutely.
So keep the Origin Center website, keep an eye on that, and thank you all for listening. And if you feel like I wish I was present in this discussion here in the audience, please next time register, because we like to have a lively discussion. So yeah, register on the website and you can be part of this audience. So with that, thank you and have a nice evening or morning or wherever you are. Bye-bye.
Bye, everyone. Thank you.
Bye. Thank you.
All right. Thank you so much.